I love cookies. I'm not even kidding. These are amazing. You know who you are. Maybe just cookie. Fantastic. It's sweet and delectable. There's going to be a little video about macromolecules. There's a bunch of macromolecules in the body. Hundreds of thousands of them, probably more, that have specific functions. They have specific jobs that they have to do. They work to make our body do the things that they do on a regular basis. Some of their jobs are to create energy. Some of their jobs are to be the energy. Some of their jobs are to hold things in, some things to hold things out. But they all have a job. They have a specific function. We have picked four. The four macromolecules that we have to deal with are carbohydrates, lipids, proteins, and nucleic acids. The last one, nucleic acids, we're going to hold off on because we're going to talk a lot about DNA. I truthfully don't have that much time to do nucleic acids right now. So when we get into DNA, we'll worry about nucleic acids. But for now, you should be taking out your macromolecules comparison table. I should take this out. So over here at the end where it says nucleic acids, this last column over here that says nucleic acids, we're not going to worry about that right now. We'll do that later. We'll talk about that when we get to DNA. That's not for now. But we are going to do carbohydrates, lipids, and proteins today. Also, don't worry about the additional information that's down at the bottom. We don't care about additional information right now. So here we go. Okay, starting off with carbohydrates. I actually introduced this with eating a cookie for a reason, because cookies are deliciously sweet. It's made of stuff. I don't know how to cook, but I can tell you, or bake for that matter, there's definitely chocolate chips in there. Chocolate chips are cocoa. Cocoa comes from a plant, and who makes glucose better than anybody, and that's a plant. So even the chocolate chips have glucose in it. What else is in there? Flour and sugar. Sugar. As a matter of fact, sugar is a sucrose, which is a 2-sugar molecule, a disaccharide, if you will, and that is a good source of energy. Carbohydrates beat everything when it comes to energy. As a matter of fact, fill that in on your chart. On your chart right there, it says functions for carbohydrate. Carbohydrates have a ton of energy. Carbohydrates are made of saccharides. That's the quote-unquote subunit. The monomer for a carbohydrate is a saccharide, and that's spelled with 2 C's, S-A-C-C-H-A-R-I-D-E. A saccharide is sugar, basically. Glucose is an example of a saccharide. When you take a bunch of saccharides or a bunch of sugars and you piece them together, create a larger one called a polysaccharide. So that would be your polymer. So we're going to do polysaccharides. An example of a polysaccharide is carbohydrates, and we have different types of carbohydrates. Plants make a specific carbohydrate, and animals make a specific carbohydrate, and they're completely different. There's 2 different ones. Just so you understand what that is, we use, it might be written backwards, I apologize if it is, but that's glucose. And so the thing that we use for a sugar molecule is really a 6-sided carbon-based molecule. It's 5 carbons. 1, 2 would be over here, 3, 4. I screwed up, but whatever. Okay, you get the idea. There's 5 carbons in the ring, and then there's an oxygen. It's the 6th one, and so it should be a pentagon. I didn't cut it so well. It's okay. So you would take glucose. Here's the glucose molecule. I'm going to put the glucose molecule on the board. Now what happens next is another glucose molecule is going to come along. So for instance, if I'm a plant and I'm physically making glucose molecules, I'm going to make one using sunlight and carbon dioxide and water. And then as soon as I'm done making one, as soon as that one's done, I need to make another one. And that's what plants do all day, every day, making glucose molecules. So now I have 2 glucose molecules. As soon as that's done, as soon as you finish making the second glucose molecule, then you're going to go and make a third glucose molecule. Now, you don't want glucose molecules just hanging out, floating around, just hanging inside the plant. And so what they do is they take those glucose molecules and they stick them together, make one big giant polysaccharide, one big giant molecule that's all stuck together that they can compact down inside the cell. And the way that they do that is like this. But that chair makes a lot of noise, doesn't it? If this side has a hydrogen and this side has an oxygen and a hydrogen, what happens is these two create a little bit of a bond. They overlap. And so what's going to happen is when this glucose molecule overlaps that glucose molecule, it is going to kick out the H, the other H, and the O. That's what that is. H2O. So a water molecule is going to get kicked out. This has a name. This is called dehydration synthesis. We'll talk a little bit more about dehydration synthesis in a couple of days. The idea here is to understand that a water molecule gets removed. And the same thing's going to happen here. The H that's over here is going to meet up with the OH that's over here. And it's going to kick that H, and then the O out. And it's going to, again, remove a water molecule, causing dehydration synthesis. I think you all know the word dehydration. I think you all know what that is. Dehydration means that you don't have water, which is why they call this dehydration synthesis. You're removing the water. Somebody's making an announcement. So there's an example of our carbohydrate. Our carbohydrate is a bunch of glucose molecules or sugar molecules that are bonded together through a process called dehydration synthesis. The elements that are involved here. A glucose molecule, just in case it's backwards, I'm going to try it this way. C6H, oh boy, 12O6. How'd I do? Something's got to be wrong. Hey, I can see it backwards. I did it. C6H12O6. So you can obviously tell from all of those things that the three elements that are involved here are carbon, hydrogen, and oxygen. C6H12O6 gives me one glucose molecule. Now here's the tricky part. If I take a C6H12O6 and another C6H12O6, you add those all together, that's C12H24O12, correct? Except for you remove a water molecule. So now you have to subtract an oxygen and you have to subtract two hydrogens. So it's really C12H what? 20? Is that right? 22. H22. And then O5. No, wrong. 12. 6 times 12 is 5. O11. Okay? So you're subtracting the water molecule from C6H12O6 and another C6H12O6 added together. So it's addition and subtraction at the same exact time. So there is your glucose. That's how you make a carbohydrate. So the basics here, as far as your macromolecule comparison chart is concerned, your monomer is a saccharide. Your polymer is a polysaccharide. The elements involved are carbon, hydrogen, and oxygen. An example of this, if this was a plant, this would be starch. If this was an animal, this would be glycogen. Sucrose is an example of what's called a disaccharide, two sugar molecules put together. Glucose is an example of what's called a monosaccharide, or one sugar all by itself. Starch is an example of a polysaccharide. Greatest function of a carbohydrate? Energy, energy, energy. That's why you have pasta parties. You have a pasta party so you can gain as much carbohydrates as you possibly can before the party or before the game or whatever it may be, before the run. Try to pop off these sugar molecules one after another. The more you pop off, the more energy you get. How do you get them to pop off? Drink water. It's extraordinarily important to drink water while you're doing a run or while you're doing a workout. And the reason is because, one, it's going to spread out the lactic acid, and two, it's going to allow for these glucose molecules to break apart. Remember dehydration synthesis? This is hydration, right? Hydration, you throw the water in, those molecules break apart, it gives you the energy that you need. Beautiful. Okay, so that's your carbs. Okay, we'll go into lipids. We'll spend a little bit less time with this, but I'm going to get rid of my glucose. Good. Moving on. Okay, next one. Sorry, let's not flip the screen like that. Okay. Next one is lipids. There's a couple of different parts of lipids. You have, first of all, you have a molecule called a glycerol. Okay. A glycerol, again, is made of carbons, hydrogens, and oxygens. Now, a glycerol is going to be the basis, the foundation, where this whole thing begins is on a glycerol. And then coming off of the glycerol, these little tails of carbons, hydrogens, and oxygens. And they're called favorite words in science, in all of biology. A fatty acid tail. Look at that fatty acid tail. Where my video goes, a little bit creepy. Okay, so normal regular old fat has 3 fatty acid tails, just like that. Okay, there's 3 fatty acid tails coming off of a glycerol. There's other molecules, other lipids that we're going to do, like a phospholipid is another example of a molecule that's a lipid. We have a couple examples right there, phospholipid. We have saturated versus unsaturated fats. Okay, this would be an example of a saturated fat. Saturated fat is called bad fat. We don't like saturated fat. Saturated fat is what causes like high cholesterol and gets all caught up within your veins and arteries and stuff like that. And the reason is because they can stack on top of each other. An unsaturated fat would be an example of one that is unsaturated with hydrogens, not enough hydrogens around. So if there's not enough hydrogens around, they can't make the nice bonds that they make normally with hydrogens. And what happens is they form double bonds. And when you form double bonds, it forms a kink. A kink in the chain causes the molecule to actually change its shape and bend and go into different directions. So this is an example of an unsaturated fat that's better for you. It's the fat that doesn't have a whole lot of hydrogens, and so it stacks up on top of it. It doesn't stack up on top of each other like it does with a saturated fat. So there's your saturated versus your unsaturated fat. There's an example of that. The words that you need there, the monomer that you need for lipids, that would be the glycerol, and then your fatty acid tails. The polymer that is created during this is not really an example of polymer, but we can call it a triglyceride. Okay, that's if you've ever heard the word triglyceride. The word triglyceride comes from tri, fatty acid tails, 3 of them, versus glycerol, glyceride. That's how they bond together. So there's your triglyceride. The elements that are involved, again, carbon, hydrogen, and oxygen. There's nothing different here. It's just the arrangement of them that really causes the difference. Your examples, like I said, a phospholipid. Phospholipid is very similar to a triglyceride, but a phospholipid has that glycerol on the top. Let me not spell it because it's going to spell backwards, I bet. That's a G. Whatever, you get the idea. There's the glycerol. And then you have 2 fatty acid tails that come off at the end. That's a phospholipid. This is extraordinarily important for the next topic, which is cellular transport. We're going to get to in a couple of days. Cellular transport involves the phospholipid bilayer. That's a massive part of it, which is why I'm spending so much time talking about it right now. Glycerol, 2 fatty acid tails. Crazy thing about this, top part's polar, bottom part's nonpolar. What? You said polars and nonpolars can't mix. Yeah, there's like a buffer area right here in the middle that keeps them apart, which causes the top part to be polar and the bottom part to be nonpolar. The top part can mix with water because it's polar. Heads like water, tails hate it. And so that's what's going to cause the phospholipid bilayer to happen, which we're going to get to in a couple of days. Then finally, proteins. Mr. Schwartzman will clean it up later. Okay, then we have proteins. Sorry, that bad boy. A protein, like I said, nucleic acids we'll save for later, but a protein. Proteins, the monomer for a protein is made of amino acids. There are 20 essential amino acids for the human body. Some of them we make by ourselves, and some of them we don't make, so we have to get them from somewhere else, which is the reason why we have to eat protein. We are meat eaters. The reason that we have to eat meat is to develop the frontal lobes of the brain. It helps our brain to function properly and also helps us to build the muscle that makes us who we are and allows us to move around. We have to eat protein. And where do we get protein from? Well, a lot of times we get it from, people say chicken or steak. That's a good source of protein. But what is chicken or steak? Are you eating a chicken, honestly? Are you eating a steak? Are you eating a cow? I don't think so. You're eating the muscle. The muscle of the cow or the muscle of the chicken is what's giving you the amino acids that you need to help develop your own muscles. So the amino acids that make up your muscles came from a chicken or from a cow or something else. Some people are vegetarian or vegan. Those people have to eat protein from somewhere else. It has to come from a legume, a nut, or something like that. It has to come from soy, somewhere else. They have to get their protein from somewhere, maybe from milk. There's plenty of places to, other than just eating chicken or steak, fish, where you can get the protein from. The proteins are made of amino acids, and there are a bunch of different types of amino acids. I did them as different colors. As you start to build your protein chain, the amino acids attach to each other, and the amino acids attach to each other in the same exact fashion as glucose molecules attach to themselves. When they attach to each other, depending on the type of amino acid, some amino acids have a charge, some of them are not charged. Some amino acids are polar and some amino acids are nonpolar. And so what happens is they create their own little attractions to each other. When they create their attractions to each other, they start to bend and shape in different ways. And that is what makes us who we are. The different types of proteins that that are required for us to work. See how it works on my shoulder? Isn't that cool? So the amino acids, the monomer is an amino acid. I'm sorry, for proteins, the monomer is the amino acid. The polymer is called a polypeptide. And these in between here, these are polypeptide bonds. The bonds between them, and that's the same exact way as before. You take the water molecule out, water goes away, and then they stick together. The elements involved, again CHO, again carbon, hydrogen, and oxygen. They're called amino acids, so there's got to be an amino group. Amino groups are the same as ammonia, which is your NH2. So you have to have nitrogen in there too. Carbon, hydrogen, oxygen. Now you have nitrogen. And some of these create what are called sulfide bonds because some parts of the amino acid sometimes, remember there's 20 different kinds, some of them have a sulfur in there. So you can also include the S. You can include a sulfur in there too. There's also phosphorus. I'm not worried about phosphorus right now because we'll talk about that later when we get into lipids. The majority of these are carbon, hydrogen, and oxygen. So far, the only ones different, amino acids have a nitrogen and they also have a sulfur in there. So, we'll do Mr. Anderson. Hope this helps. Fritsche signing off. No?